friends, I am Dr. Priyanka Shrivastava. I am assistant professor, and today we will be studying the prokaryotic transcription. So, if we go for the definition, the cellular process in which the RNA is synthesized using DNA as a template is known as transcription. RNA is a polymer of ribonucleotide held together by 3' to 5' end phosphodiester bridge, and they are single-stranded. So it's very important to understand the difference between the DNA replication and RNA transcription. Replication, it is a process of the formation of the two copies of the DNA molecules. Entire genome is copied. But in transcription, only a segment of the DNA is copied to the RNA. It is the synthesis, then the replication, which is a synthesis or the formation of DNA from the DNA. But transcription, it is the synthesis of the RNA from the DNA. DNA replication, both the strands of the DNA take part in the replication. But in transcription, only one strand of the DNA acts as a template and get transcribed into the RNA. DNA replication, the nitrogenous bases found in the DNA polynucleotide chain are adenine, thymine, guanine, and cytosine. Nitrogenous bases found in the RNA polynucleotide chain are adenine, uracil, guanine, and cytosine. DNA replication is catalyzed by the DNA-dependent DNA polymerase enzyme. And in transcription, it is catalyzed by the DNA-dependent RNA polymerase enzyme. So if we talk of the prokaryotic RNA polymerase, it has subunits, that is the alpha. There are two subunits of the alpha, beta, beta dash, omega, and a sigma also. So the beta dash, it is the largest subunit and consists of the active center that is responsible for the RNA synthesis. Beta, it is the second largest subunit that consists of the remaining portion of the active center. Alpha, it is third largest subunit that is present in two copies, as I told you earlier. And the omega, that is the smallest subunit that facilitates the assembly of the RNA polymerase. Then there is all the 5 units make the core enzyme, and when a sigma subunits come and join with the core enzyme, it forms the holoenzyme. So the prokaryotic RNA polymerase holoenzyme consists of alpha 2, beta, beta dash, sigma, and prokaryotic RNA polymerase core enzyme consists of the alpha 2, beta, beta dash, omega. Okay. Now coming to the prokaryotic promoter. Promoter consists of two conserved sequences, that is the -35 and the -10 element. -10 region, it is also called the Tata box because its sequence is TATAAT, and -35 region has the sequence of TTGACA. These sequences are upstream to the site from where the transcription begins. Binding of the holoenzyme to the two conserved sequence of the promoter forms a closed complex, which is very much required for the RNA transcription. Coming to the 3 stages of the prokaryotic transcription, we have an initiation phase where the RNA polymerase recognizes the promoter and starts the transcription. Then elongation phase, the RNA strand is continuously growing and making, and lastly but not the least is the termination phase where the RNA polymerase stops the synthesis of the nascent RNA and the RNA is separated from the DNA template. So coming for the first, that is first of this, the initiation of the transcription. Closed complex, binding of the RNA polymerase holoenzyme to the promoter sequence forms a closed complex. Then comes the holo-open complex, where after the formation of the closed complex, the RNA polymerase holoenzyme separates 10 to 14 bases extending from -11 to +3, which is called the melting. So the open complex is formed. This changing from the closed complex to the open complex is also called the isomerization. Finally, we have a ternary complex or the tertiary complex. RNA polymerase starts synthesizing nucleotide. It does not require the help of the primase. If the enzyme synthesizes short RNA molecule of less than 10 base pair, it does not further elongate, which is also called abortive initiation. This is because sigma 3.2 acting to mimic of the RNA, and it lies in the middle of the RNA exit channel in the open complex. When the RNA polymerase manages to synthesize RNA more than 10 base pair, it ejects the sigma 3.2 region and RNA further elongates and exits from the RNA exit channel. This is the formation of the tertiary complex. Coming to the elongation of the transcription stage, after the synthesis of the RNA more than 10 base pair, the sigma is ejected and the enzyme moves along the 5' to 3' direction, continuously synthesizing RNA. The synthesized RNA exits from RNA exit channel. Newly synthesized RNA undergoes proofreading by hydraulic editing. And last stage is the termination of the transcription. There are two mechanisms, row independent and row dependent mechanism. In the row independent mechanism, where the transcription is terminated due to specific sequence in the terminator DNA. The terminator DNA contains an invert repeat which causes complementary pairing as the transcript of the hair RNA forms a hairpin structure. So it is the hairpin structure which is responsible for the termination of the further RNA synthesis. Second is the row dependent. In this mechanism, as you can see from the diagram, that transcription is terminated by the row protein. There is a row protein which binds to the RUT site and then scans back to the part of where it found the DNA RNA hybrid, and once it forms the DNA RNA hybrid at the RNA polymerase site, where it gives the signal to stop the transcription, to stop the further synthesis of the nascent RNA. So this is all about the prokaryotic RNA transcription. So students, you have seen how the prokaryotic RNA transcription take place. There are two important parts. One is the enzyme, the part of the enzyme, and second is the stages of the transcription. So it is very important to understand both the things. First is the enzyme, that are different parts of the enzyme, and the three stages of the transcription. So that is all about the transcription. We will see you again. We will meet again with the next some new exciting topics of the science.